which is great to see. And as everybody can also see, we've started recording, so just want to highlight that in case anybody doesn't want to be recorded. Hi everyone, my name is Ben Corrio. I'm one of the HP project leads here at SEARCH, the Centre for Excellence in Rural Sexual Health. I work out of Wangaratta primarily, and I live in Yakandanda in northeast Victoria. It's my pleasure to welcome you all to SEARCH's Sexual Wellness Professional Development webinar for February 2024. We host PD webinars monthly for most of the year, so I would strongly encourage you to check out our website and keep your eyes peeled for our bi-monthly newsletter for news about future webinars. Before we go any further, I would, of course, like to offer an acknowledgement of country. On behalf of SEARCH, I'd like to acknowledge the Yamatang people who are the traditional custodians of the land that I'm speaking to you from today. And I, of course, also acknowledge the traditional custodians of the lands on which each of you are living and working today. We extend this respect to elders past and present, and of course to all Aboriginal and Torres Strait Islander colleagues joining us today. It would be great if you could introduce yourself in the chat, and if you feel up for it, letting us know whose country you're calling in from. While everybody does that, it's my pleasure to introduce Associate Professor Fabian Kong, who will be our speaker today. Fabian is the Deputy Head of the Sexual Health Unit at the University of Melbourne. He's a clinical pharmacist and sexual health epidemiologist currently working to optimise treatments for STIs, particularly for multi-drug resistant gonorrhoea in the mouth. This includes clinical trials to collect drug data or pharmacokinetics from the oropharynx to optimize treatments using modeling and the development of the world's first oral human tissue model of gonorrhea infection to screen emerging treatments to treat oral gonorrhea. He's also working on developing a fully automated online STI clinic in Victoria. So just very briefly before I hand over to Fabian, a little bit of housekeeping. We do anticipate having time for questions at the end. So please save them for the end. If you'd like to drop questions in the chat, I can sort of collate questions and hand them on to Fabian. We can also open them up for the floor to offer questions too. Just reiterate, we are recording this session. So if you'd like for any of your questions to be anonymous, please note that when you drop them in the chat, and I'll sort of reiterate them as being from an anonymous source. So without any further ado, Fabian, over to you.
Hi everyone, I'll just share my screen. There we go, you should be able to see that. So I will just get started. First, I'd like to acknowledge the traditional owners of the land that we're meeting and pay my respect to elders past, present, and emerging, and welcome everyone that is here. So today I'm going to talk about this topic about optimizing STI treatments using pharmacokinetics with a focus on infections of gonorrhea in the mouth. Oh yeah. So today I'll talk about the treatments that we've had for Neisseria gonorrhea or gonorrhea, and talk about the limited options we have now, and then talk about the treatments we've had in the past, what we have now, and also what's emerging in clinical trials with a lens of pharmacokinetics. So I'm going to focus on oral infections because the cures for oral infections are 20% lower versus other infection sites, such as the rectum or the genitals. We've already seen resistance to all classes of antibiotics to gonorrhea. You get ongoing transmission by oral sex and saliva. And most importantly, oral infections is where it facilitates antimicrobial or antibiotic drug resistance development. So it's really important for us to cure oral infections, otherwise AMR starts to spread. So I'll talk about STI optimization using pharmacokinetics or PK, with a particular importance on getting data from tissues or fluids at the site of infection and not just the blood. And I'll discuss this other thing called pharmacodynamics or PD, which essentially is a measure of efficacy for treatments. So what I want to talk about first is obviously things like the treatment efficacy. We know now that treatment efficacy can actually vary by different sites of infection. So I'm just going to give a quick background on gonorrhea prevalence. So oral prevalence of gonorrhea sits around 2 to 10%, lower in heterosexual populations and much higher in gay and bisexual men. And rectally, we see about a prevalence of 2 to 6% in those similar populations. All studies have shown that in terms of oral gonorrhea, this is much more difficult to cure versus genital infections. And an old review found that overall gonococcal cure rates were sitting around 96%. And then if you look at oral infections in men, it's much lower, sitting around 80% versus females about 84%. And we know also for other infections at other sites, such as chlamydia, that 2 randomized controlled trials, including the one that we undertook, that azithromycin is now 20% less efficacious compared to doxycycline. And if we look at kind of the tissue level data, we know that drug levels to inhibit or cure chlamydia using azithromycin, you require 4 times as much in colorectal tissue versus endocervical tissue lines. And so based on this, we now no longer recommend azithromycin for treatments of rectal chlamydia. So I just want to give a quick summary since I'm talking about gonorrhea and oral gonorrhea and effectiveness. And I just want to present some results of some efficacy. So treating rectal chlamydia, we undertook a meta-analysis of 54 studies, 40 regimens, and just over 1,800 patients. And we found that this meta-analysis found that ceftriaxone-containing regimens were the best, with efficacies at 100%. We found that there was no difference in whether you chose to use single or dual therapy. So this is ceftriaxone with things such as azithromycin. And these list of drugs here are the drugs that are now emerging in clinical trials as what might become the new treatments for gonorrhea. And I just want to highlight these 2 in bold, gepotidacin and zoliflodacin, just because they're the ones that are going to hit the market soon. The other 2 on the top are no longer relevant because they've been removed off the market. So if you look at that table at the right on the right there, these are the drugs I'm talking about: zoliflodacin at 2 to 3 grams, and gepotidacin at 1.5 to 3 grams. And you can see the efficacies there are sitting around 100% for treating rectal gonorrhea. And note that the lowest confidence intervals are below 90, and criteria do require that lower confidence interval needs to be at least 90% for it to be considered effective. And we get in those low confidence intervals because the sample sizes are so small. So these are the number of patients that were studied to get those efficacy measures there. And also these treatments have got much higher side effects versus current treatments, and I'll discuss this later. So in terms of oral gonorrhea, these are the current efficacies of current treatments. So again, we undertook another meta-analysis showing lower efficacy in the mouth versus the rectum. These studies were in 9 studies, 19 regimens, and just over 450 people. So you can see overall ceftriaxone regimens are the best, but they're much lower at 98%. Similar single or dual therapies have no difference in terms of efficacy. And again, in terms of emerging treatments, zoliflodacin is again the one that we're going to focus on here. And again, very small sample sizes. So if we look at the emerging treatments here, zoliflodacin at 3 grams, studied in 9 people, showed efficacy of oral gonorrhea cure about 78%. But look at gepotidacin at 3 grams. This is the one that's also going to appear in the market in the next few years. Studies in 2 people and efficacy of 50%. So there's still lots of work to be done with these 2 new emerging treatments to cure oral gonorrhea. So the one question that will arise is these issues that we have with low sample sizes, and that then results in these potential candidates having lower confidence intervals of less than 90. Most of these studies don't randomize by the site of infection. And the major issue we have now is that these drugs may not treat gonorrhea at all infection sites. So take the scenario here where you get gonorrhea infection in the rectum and in the oral space. These are low sample sizes. So if you get dual infection at those both sites, you give them zoliflodacin 3 grams, you get 100% cure in the rectum. But if you give that in the oral, you only cure 80% of oral infections. Similarly with the other drug that's coming on the market, using gepotidacin 3 grams, really good at curing the rectum, but not good at all in treating the oral. So we're getting this dilemma at the moment that current treatments in the future might cure one side of infection, but on another, and oral infections are the one that's going to be majorly disadvantaged. So I'm going to start going into these PK/PD measures. So the definitions here are that pharmacokinetics. This is the effect of the body on a drug. So you administer a drug, the body absorbs it, distributes it, metabolizes it, and eliminates it, or ADME. And then, so in this presentation, I will talk about the distribution of these drugs at non-genital sites. So particularly the oral tissue or fluids, including saliva, gingival fluid, and also some mentions of the rectum as well. And pharmacodynamics, or PD, this is the effects of the drug on a body, or in this case, an organism. And in this presentation, consider pharmacodynamics as the efficacy of a drug against gonorrhea. And CDC considers for gonorrhea that an effective treatment must be at least 95% effective, with that lower confidence interval being at least 90. So the question is, how can efficacy be improved by knowing these PK/PD measures? So things will start getting complicated now, but just hang in there. I'm going to try to explain it very clearly and slowly. So this is your standard kind of PD, PK modeling. graph when you take a drug. So when I'm talking about drug levels, I'm talking about free drugs. And what I mean here is that these drugs are free because they're not bound to protein. So these are unbound protein drugs. So, and free drugs are the pharmacologically active drug, and that's the drug we tend to want to measure. So this is your kind of like a graph about when you take a drug. Here's the concentration of that drug in your body or tissue, and this is over time. So, you take a drug, concentration goes up, comes back down. So I'm going to just give you some of the major components to consider. So this one is really important. It's called the MIC, or the minimum inhibitory concentration. So this is the lowest concentration that you need at a tissue to inhibit the growth of gonorrhea. And the lower the number, the better, because and the other one is this other one at the top called the maximum tolerated concentration. So you don't want your drug concentrations to go beyond this, otherwise you're going to get unnecessary side effects in people. And in between these two ranges is the therapeutic range. So let's start it. You get given a drug, the concentration goes up and it reaches the maximum concentration. This maximum concentration is called Cmax, or maximum concentration. And the time that this occurs is called Tmax, or the time to maximum concentration. So you take that drug, it hits the maximum concentration and starts to go down. So you can see here, when that drug concentration goes up, it goes beyond this MIC. So here is where you start getting a therapeutic effect, because the concentration is now above the MIC, comes back down, still remains above the MIC, and then drops back down. So this amount here above the MIC is what we consider then the duration of action, or the time above MIC. And the last one is this thing called area under the curve. And as the name suggests, it's this shaded area under the curve. And this area under the curve just represents the total amount of drug that's absorbed in the body. So these things are really important when I further talk about these concentrations, but the MIC is the really thing that you need to keep in mind. The lower the MIC, the better, because all the measures of PK and PD all are relative to the MIC. So I'll keep re-emphasizing this as I present along. So the key pharmacodynamic measures of efficacy are all relative to MIC, as I mentioned before. So you get these three major type of PD measures of efficacy. One is the time of that free drug concentration above the MIC, or we see it as T above MIC. And we term this time-dependent killing. So this is really important, because what this means is that you need the duration of exposure of that bacteria to antibiotics is really important for these antibiotics to ensure cure. And these two drugs, ceftriaxone and capsaicin, are some examples of drugs that are appropriate for this measure. The other one is this area under the curve at 24 hours. Okay, so that's what I'm talking about. So again, remember, this is the MIC, this is the time above MIC, and that's the duration. This is what I'm talking about here in this one. The next one is the area under the curve at 24 hours relative to MIC, or this abbreviation. And this is what we term a concentration dependent. And with these type of drugs, it's the total dose that you give them that's more important. And it doesn't matter how you give that total dose, it's just the total dose that you give. And these are the drugs that are most appropriate for this type of measure. And azithromycin is the one I'll talk about. So again, if we look at the graph that we had before, concentration goes up, goes down. At the 24-hour mark is here. It's all this shaded area under the curve, and that's related to MIC. And the last one is called the maximum concentration to MIC, Cmax to MIC, and this is concentration dependent, and these are the drugs related. So here is just this related to MIC. But in discussion, I'm just really going to talk about these two, particularly with ceftriaxone and azithromycin, since these are the ones that we're going to use for treatment. So I hope that's clear. I'll keep going. So one of the key questions is, how long does gonorrhea need to be exposed to antibiotics to cure? So the CDC says for gonorrhea efficacy, for a penicillin, the serum concentration needs to be at least 4 times the MIC for at least 10 hours after you reach the Cmax, or the peak concentration. But unfortunately, these papers are from 1960s. It's based on blood, penicillin, and at the urethral site. And so what does this do? We can't really use this measure for tissue levels, for non-penicillins, and for other sites of infections other than urethra. So in terms of ceftriaxone, which I alluded to before, T above MIC is the measure of efficacy. So lots of modeling has been done now, and studies have shown that for ceftriaxone, the time above MIC needs to be more than 70% of the dosing interval. So what that means is, if your dosing interval for ceftriaxone, which is one gram every 24 hours, 70% of 24 hours is 17 hours. So what this modeling suggested, that for ceftriaxone to work against gonorrhea, you need that time above MIC needs to be at least 17 hours to work. And in fact, if you look at other studies, this is what we show. This is what we see, that 0.5 gram and 1 gram of ceftriaxone is effective for 33 to 41 hours for gonorrhea infections, for gonorrhea strains that have a low MIC. But again, this is based on blood levels, and we don't know what these numbers will represent for tissue. So we can use PK/PD model to optimize present and emerging treatments using these kind of methods. Heavy table, but don't worry about it. I'm going to explain it to you. So you can imagine that when you take a drug, the amount of drug that penetrates a tissue is really, really important because that's going to go to the site of infection. So this is a table of antibiotics, some of the kind of basic pharmacokinetic measures, like how long it takes to meet concentration, the half-life of how long it hangs around. But I want you to focus on this thing called the volume redistribution. The volume redistribution, as the name might imply, is the volume, the kind of theoretical volume that a drug distributes in the body. So a drug might distribute in 1 liter of body, and another one might have a volume of about 10 liters. So that drug that has a volume of 10 liters or distributes in the 10 liters has a greater tissue penetration. And these are the various volumes of distribution for these antibiotics, but I'll get back to that. So I want to give you a benchmark. So there's a drug called warfarin, which is a blood thinner. And this drug here mainly stays in the blood and has very little penetration into tissue. And for a drug like that doesn't penetrate tissue and mainly stays in the blood, the volume redistribution number is 0.14. So any volume redistribution that sits around that number means that it doesn't really penetrate tissue and just stays in the blood. So if we go back to the table now, you can see that ceftriaxone and gentamicin don't really penetrate tissues very well. And I want you to keep an eye on this gentamicin because gentamicin doesn't penetrate tissue very well. And later I will show that's probably why we see such poor efficacy of gentamicin in tissue and oral sites because it doesn't get there. If we compare other drugs such as Pepixim and Cipro, you can see that the volume distributions are much higher, so greater tissue penetration. And look at azithromycin, massive, and then doxycycline even better, even bigger. So understanding some of these PK information allows you to understand whether or not that drug is likely to penetrate into tissue and therefore effective at sites of infection. And these things are measured using PK information. So now moving on to kind of tissue concentrations at these extragenital sites that I'm focusing on. So the rectum or the mouth, and these are the antibiotics that we use. So currently, in terms of rectal infections, we have very little data on these antibiotics and whether they distribute into rectal tissue. So we don't know how well these drugs would work against rectal gonorrhea. But we've undertaken a study to show that for azithromycin, given 1 gram of that, you get about 5 times as much azithromycin in rectal tissue relative to blood. So it's got good penetration to rectal tissue. And doxycycline, similarly, we see studies sitting around 2 to 4. So they have great penetrations. And then if we look at the oral space here, we're looking at that green part, and we look at the concentrations in different oral spaces compared to plasma. So the ratio of tonsil to plasma, the ratio of drug in saliva to plasma, and then the ratio to gingival crevicular fluid, or this is the fluid that you find in gingiva. You can see that ceftriaxone, very little distribution into the tonsils. You get 20% in tonsil tissue relative to blood, very little in saliva, no information on gingival fluid. And this can vary, for example, with azithromycin, which just penetrates in every tissue space, 150 times more azithromycin in tonsil relative to blood, and 6 to 40 times more in saliva and other fluids. And this can vary then with other drugs here, ciprofloxacin, doxycycline, high concentrations in tonsils, less so in saliva, huge amounts in gingival fluid. So having these type of studies, then again, particularly in spaces like the oral space or the rectal space, where you plan to use these drugs for these areas of infection, the information is really important before we go into trials. And to complicate things even further, pH can also reduce efficacy. So the pH can affect this thing called drug ionization, and drug ionization is how much electrical charge there is on a drug. And drugs that are unionized or don't have a charge, they're the ones that are able to penetrate into cells and are pharmacologically active. All drugs that I'm speaking about for STI treatments, except for the cephalosporins, have a reduced efficacy when you reduce the pH. So for example, when you drop the pH from 7 to 5. And this is really important because pH varies between different tissue sites and whether or not there's a disease state within those tissue sites. So I've got this figure here, pH acidic on this side, really alkaline on this side. And you can see here the various pH in these various tissues. We know already from studies that with the vagina, you need a pH of less than 4.5 to be healthy. And if it goes above that, you get things like bacterial vaginosis and things happening. But to complicate things even further, the mucus that sits on top of the vagina, it's a much higher, it's on the other side. So when you're looking at drug distributions in the vagina, you need to consider both of these different spaces. Urethra is down this end at the more acidic end. The oral space is sitting at more at the alkaline end. And with the rectum, it's even higher, particularly if you have rectal STI, the pH goes quite up at all. So what we try to do, so in my PhD and my further studies previously, we try to understand why azithromycin in rectal chlamydia treatments was failing about 20% more, 20% greater failure with azithromycin versus doxycycline when we use that for rectal chlamydia treatment. So here we looked at various studies looking at azithromycin in rectal tissue, which was a past rectal chlamydia treatment. We know from the literature that macrolides are ineffective when the pH was below or equal to 6, and the optimal pH actually for macrolides is about 8. So we undertook a pharmacokinetics study where we gave men 1 gram of azithromycin, and then we measured levels in their rectum. And in one patient, we found 10 times higher azithromycin in rectal tissue who was on chronic esomeprazole. And esomeprazole is a drug that raises gut pH. And in another study in Amsterdam, which we took the rectal mucus of patients attending a sexual health clinic. We took that mucus and then measured the pH to see what the rectal pH might be and see how that might affect treatment failure. And we found in this study that 20% of MSM, or men who have sex with men who had a rectal STI, had a rectal pH of less than 6. And this is the pH that we would expect failure for azithromycin. And this equated to about the 20% treatment failure we see in RCTs of azithromycin versus doxy. So the pH stuff is interesting for me. I'm not sure what you can do with it. You potentially might be able to use pH at a clinic level to measure, for example, saliva pH and see whether or not that might predict failure. So pH for me is a theoretical thing, and it's something I don't know whether we could be able to practice in clinical clinics. And so this is just another example. If you reduce the pH, you can reduce the efficacy, and this reduced efficacy is shown by an increase in MIC. So here's this table that if you reduce the pH, increase in MICs, if you reduce the pH from 7.3 to 5, these numbers here, the number of fold increase in MIC. So you can see here for these drugs here, if you drop the pH, MICs can increase up to 1,000 times for azithromycin, up to 4 times and 8 times for your quinolanes, and gentamicin 32. So reducing pH is no good, but didn't seem to be affected by penicillins at all. And if you look at Cefoxitin, it's the opposite, that if you increase the pH, this makes it worse. And for me, Cefoxitin is really important, and we're doing some studies on this now because it's the kind of the only oral treatment that we have with ciprofloxacin, and it's really important for this drug to be effective because it's used in resource poor settings where injections aren't possible because the staffing aren't there to provide those injections. So in this study here, we show that increase in pH from 5 to 8 can increase the MIC 2 to 4-fold. So here's your pH, goes from pH 5, pH 8. These two, you see a 2 times increase in MIC. And for E. coli, we see an increase in 4 times in MIC. And these pHs can change obviously in disease states. So the other thing to consider for the mouth is that the mouth is an extremely dynamic environment. Those epithelial cells of the mouth, so those cells that sit on top of the surface of your tongue and cheeks, they're replaced every 3 hours. So if you've got drugs sitting in those epithelial top layers and it's constantly being replaced every 3 hours, that will therefore have an impact on the time above MIC that might have in tissue. We also know that most of the bacteria that you find in saliva is actually bound to epithelial cells. So the bacteria infects the cell, it dislodges and then floats in the saliva with the epithelial cell. So it's actually unsure what saliva levels might be and might be less effective than actually levels in tissue. And also you've got this constantly moving fluid. You get the saliva flow rate, you get about 20 mils happening in an hour. You get gingival fluid, which kind of produces small amounts every hour. And this is increased with disease. So your mouth is this really dynamic space, lots of tissue being replaced, lots of saliva flowing and flushing your mouth. And I think this is one of the reasons we see complicated failures in the mouth, because drug concentrations need to be really, really deeply penetrating in tissue for it to be really effective. And also in the mouth, you've got different types of tissue. So different tissues in the mouth. And for example, the back of the throat, that type of tissue there is kind of similar to what we find in genital tissue. And so you then have to understand whether or not the drug distributes differently in tissues, different tissue types. But for us to understand this, we're battling with this question about, if we don't know where gonorrhea grows in the mouth, then how do we know that we need to target those areas in the mouth with antibiotics? So the first thing we needed to know was, first we need to know where gonorrhea grows in the mouth. And so what we've done is we started doing some lab studies to understand where that is, just so we understand where drugs need to be delivered to be effective. And so what we found in these studies, which I'll show later in our results, that tonsils had the highest rates of infection, but all sites in the mouth were infected. And we looked at this in four different human tissue types: the gingiva, floor of mouth, cheeks, and tonsils. So all these sites were infected, but lots of infections in the tonsil. So here we talk about collecting. So in our studies, we've collected samples from the tongue, these tonsillar pillars up here, and then the back of the throat to kind of represent the various tissue types in the body. And I'll give you some results later. So we know gonorrhea now is a global threat to human health. I'm going to try to remove this. And we know that over time now, resistance to gonorrhea has occurred over time, meaning that the amount of available treatments now have become less and less and less. And what that means that if we've run out of treatments now, it's really important that we optimize the drugs we have now, because the number of drugs that are emerging in clinical trials and the new drugs that we have available to us is basically nil. So I'm going to start talking about past treatments for gonorrhea and present ones and look at how the difficulty in having treatments in the marketplace at the moment. So these two drugs here, gadafloxacin, given at those doses, was shown to have 100% cure rates for the rectum and pharyngeal site, but this was removed off the market in 2008. There's another drug called gemifloxacin, which was given with 2 grams of azithromycin. Again, they're effective in curing rectal and pharyngeal infections. But in fact, the effect here is actually from the azithromycin, not from the gemifloxacin, because studies have shown that azithromycin 2 grams by themselves can cure 100% of oral gonorrhea. But again, this drug is no longer available on the market. So what some Netherlands colleagues did in 2022 was do this trial called the BIGO trial, which they looking at drugs that are currently available on market, and if we give single doses of these currently available drugs, what the efficacy might be for treating genital and oral gonorrhea. And I'm just going to present the data from the oral gonorrhea. So these are the 3 drugs they treated. Ertapenem, 1 gram intramuscular, had an 88% effectiveness against oral gonorrhea. Gentamicin, 26% cure rate, very, very low. And fosfomycin, given 6 grams orally, didn't do anything at all. So why didn't they work? And I'm going to start applying some pharmacokinetic lenses to this. So firstly, ertapenem, effective at 88%. We know that that's really effective, but it's really very unlikely that ertapenem is ever going to be used as first-line treatment. And in fact, we use it now to treat multi-drug resistant gonorrhea at 1 gram daily for 3 days, simply because ertapenem is a broad-spectrum antibiotic and it has its own carbapenem-resistant issues in hospitals. But look at gentamicin. Gentamicin was only effective in clearing 26% of oral gonorrhea. And this is likely because of poor tissue penetration. So if we go back to the MICs that is required for gentamicin to cure gonorrhea. So the MIC for gentamicin for gonorrhea is this number here, 4 micrograms per gram. So that's the level that you need to get in tissue to kill gonorrhea using gentamicin. And if you compare it with ceftriaxone, it's much, much higher. So you really need lots of concentration in gentamicin to hit tissue. But I'll refer you back to that other graph that I had before, gentamicin and the volume distribution. The volume distribution was this number, and we know that that's the number where the drug just stays in the blood and don't penetrate the tissue. So it's likely here that because gentamicin doesn't penetrate into tissue very well, which is why we've seen this poor tissue penetration, which is probably why we've seen such poor effects in oral space. And with fosfomycin 6 grams, no effect, but most people got diarrhea from it. So this type of treatment is not going to be very user-friendly. And so the question would be whether you can reduce and split these doses. So they've previously found that if you give 3 grams instead of 6 grams, but you give that over 3 days, this is able to cure lots of genital gonorrhea. So in fact, drugs like fosfomycin might actually be better for genital infections rather than the oral infections. And so this starts to hint about this idea that I think we need to start moving away from single doses for treating STIs, because you may not cure all the bacteria there, and the bacteria that is left behind is likely to generate resistance and then promulgate. So can we optimize gentamicin for oral gonorrhea? The answer is probably not. We know that for oral gonorrhea, gentamicin can't be used as monotherapy. 360 milligrams only cure 20% of oral infections in men. But when you combine it with azithromycin, you saw a great cure. But again, it's the azithromycin doing the hard work and not the gentamicin. So I think that gentamicin is only really going to be good for urogenital infections as part of dual therapy. And why do we think that's really good for genital infections? So it's really based on the same theory that you use for urinary tract infections, that you want an antibody that's excreted in the urine and found in urine to cure UTIs. And similar for urogenital infections, gonorrhea, we know that gentamicin, more than 70% of gentamicin is found in the urine within 24 hours. And this is probably why it works so well for genital infections. And if you combine 240 milligrams with azithromycin, you see a cure rate of 94% in genitals, 90% in the rectal space, and 80% in oral. But gentamicin is not particularly a nice drug, lots of kidney toxicity, potentially also ear toxicity as well. So I'll go back now to about the struggles we have now in finding new drugs for gonorrhea. So there's not many new drugs available, and particularly they're not very good for oral gonorrhea. So I previously alluded to these three here. So what I'm going to do here is present the drugs that would have been available from the highest efficacy to the lowest efficacy. So solithromycin had the highest efficacy against oral gonorrhea, 95%, 94%, but this was removed from the market due to liver toxicity. The next most effective is delafloxacin at 89%. Phase 3 trials will also stop. So only these two drugs that we have here highlighted in red. So zoliflodacin here, it's about 78% efficacy against oral gonorrhea, given as a 3-gram single dose. And in phase 2 trials, we found that this dose of 3 grams gave 10% of gut side effects for people. But in fact, if you use modeling using hollow fiber infection models, which I'll talk about later, it actually suggests that you need to give 2 3-gram doses rather than just a single dose. And the other one that's going to hit the market probably in the next few years is gepotidacin, poorly efficacious against the mouth at 50%. And they're kind of do multi-dose trials now where they're giving 3 grams, then another 3 grams 10 to 12 hours later for urogenital infections, and we're waiting for the results of that. But if you give 3 grams as a single dose, which is what they use here for this oral infections, a third to 1 in 5 people would get diarrhea and abdominal pain. And again, modeling has suggested that 3 grams twice a day might work better. So in fact, these two drugs that are about to hit the market, they're all going to be marketed for single doses, even though modeling suggests that 2 doses is better. And lastly, because we've run out of treatments, there's been other calls to use other new drugs that might be beneficial. There's a drug called lefamulin, which they could use for multi-drug resistant gonorrhea, and it might also be effective for other STIs such as mycoplasma genitalium and chlamydia. But this company shut down last year. And there's another drug called this one, which I won't try to pronounce. It's a new intravenous drug, which is for a new gram-negative antibiotic. But the issue with this drug is you have to give it as a 3-hour infusion, and you need to give it 3 times a day, which is therefore not very useful. And the reason why you have to give this drug as a 3-hour infusion is because the time of MIC is only met when you give this infusion over 3 hours. So I want to go back to this idea about single doses. So single doses, comes back on. So single doses of gonorrhea is associated with high side effects. So we see that with drugs such as ceftriaxone, we get lower side effects. So here with ceftriaxone monotherapy, you see fairly lowish kind of side effects. The highest ones we see are from emerging treatments and dual therapy. If you look at azithromycin 2 gram as monotherapy, lots of people get side effects. If you look at emerging treatments now or using dual therapy with azithromycin, you see lots of side effects. You get about 1 in 3 to 1 in 5 people always having a side effect. So the one way to deal with these high side effects with single doses is potentially to split those doses. And you can split doses with drugs that are concentration-dependent drugs. So the ones I mentioned before, which are AUC to MIC numbers, and these such drugs such as azithromycin, gepotidacin. So I'm going to give an example next about how you can split doses of azithromycin, knowing that it's this type of drug. And also, if we could have also split gepotidacin doses to reduce side effects. So with these types of drugs, AUC to MIC drugs, the total dose is really important. So it's important to give 2 grams of azithromycin, for instance. And that's more important than how you might be giving an example 1+1. So taking back to trying to give some more practical examples, so how can we optimize azithromycin, which is a concentration-dependent drug? We said before that that's the PD measure. Total doses are important, and we can actually optimize its side effects by adjusting the delivery of that total dose, and that is splitting the dose. So we know for gonorrhea, if you give 2 grams of azithromycin as a single dose, 70% of people are going to vomit. But if we split the dose to 1+1, and the second dose given 6 to 12 hours later, we can actually reduce the nausea by 1/3 and diarrhea and vomiting by about 5 to 10%. So in this study that we undertook to understand this, we didn't get a lot of follow-up from people using the split dosing, so we didn't really understand whether the efficacy was. But there was 3 patients that did return using the split dose regimen, and we found that there was a reduced side effects, and it did cure the oral gonorrhea. So there is this ability that you can split doses of antibiotics to maintain its efficacy, but then reduce its side effects. And in fact, this recommendation is now what WHO recommend for people who want to use 2 grams of azithromycin just to reduce the side effects, but not affect efficacy. But having done all that work, azithromycin is going to be removed from dual therapy treatments for gonorrhea anyway. So this is kind of more academic now. And then if we then look at azithromycin, ceftriaxone, which is time-dependent killing. So again, time of free drug event, MIC, you can actually optimize this by increasing the duration of exposure. So you want to increase the exposure time for gonorrhea to the drug. So we know historically from the 1990s, we used 250 milligrams of azithromycin versus ceftriaxone to the 1 gram stat. Now we see the 2020s, and it's very likely now that 1 gram monotherapy is likely to be standard, except for the US, where if you're less than 150, they give you half a gram. So if we look at how this works again, remember this was the MIC. This is the concentration you need to hit before you get therapeutic effect. You give a drug, it comes down, and so this time here above MIC is your time above MIC. But if you give a higher dose and this comes down, then your time above MIC is much longer. So that's a theoretical basis of kind of optimizing drugs which have this time above MIC. And so there's various ways that we can also optimize treatments. And so I mentioned before about the trials suggesting that double doses were better than single doses for gepotidacin and zoliflodacin. And so there's this model called hollow fiber infection model. And essentially what this is trying to replicate the body in a lab. So you get all these tubes that represent blood vessels, and these tubes then go into a cylinder, and surrounding these tubes is a medium, and in this medium they stick gonorrhea in there. So this is kind of trying to replicate the body. And what these hollow fiber infection models do is they try to inform doses that you could trial in clinical trials using PK/PD data to say in the trial, if we try these various new doses, will that then improve treatment? And then you run them in clinical trials, and then you see what the effects are. Obviously, they're quite complex. Look at the picture. They're very expensive to run. They cost about $250,000 a year to run per drug, and they take about a year to complete. So this is a great model, and the only people in the world that does it is WHO, one of our collaborators. And then the only lab actually in the world that has the quality assurance to run gonorrhea in these models. So not very many people can do these studies. And the other model is that one that we're developing now, trying to understand where gonorrhea grows in the mouth. So where do drugs need to be distributed in the mouth where gonorrhea grows? Currently in the literature, all the distribution of drugs is limited to the tonsil. So what we've done now is we've developed this 2D model of oral gonorrhea infection using human oral cells to try to understand what the oral gonorrhea infection dynamics are. So this is what we've done. So we've chosen these various tissues here. And so these tissues start from the front of the mouth to the back of the mouth. So from the front of the mouth, you get the gingiva. And as you move to the back of the mouth, you get the floor of the mouth, the cheeks and tonsils. So what we did is we looked at these human oral cells, and then we infected them with gonorrhea. And then we looked at this thing of how much gonorrhea or CFUs, or how much gonorrhea then infected or invaded these cells. So when we looked at using a sensitive strain of gonorrhea and infected it in these cells, and this is the FA1090 cell, we found here that the cheek and tonsil cells were heavily infected with gonorrhea, but it was still infected in these other sites. But then if we looked at using trialing and infected these cells with a resistant strain of gonorrhea, H041, we found this massive infection in tonsils and in cheek. So what this tells us in the model is that gonorrhea infects all tissues, and that the drugs that we provide to cure oral gonorrhea need to also target all these different parts of the mouth, particularly in the tonsils where we get high invasions of gonorrhea in those tissue types. And this is just a picture of our model. These are the oral cells that are in blue, and these green things are the gonorrhea and stuff. So by looking at these kind of confocal microscopy shots, we can actually see whether gonorrhea is sitting on the surface of an oral cell or whether it's invading inside the cell, as you can see here as well. So this is kind of looking at how gonorrhea invades the cell and therefore the infection dynamics. So you can also do clinical trials. So we've got a clinical trial which had finished recruitment last year, where we randomized 20 men to 3 treatment arms. The 3 treatment arms being these 2 monotherapies of ceftriaxone and cefixime. And then the dual therapy that we tested was ceftriaxone with azithromycin, but using the split dosing of azithromycin. And what we've done, we give these men these treatments, take samples in their mouth, saliva, and oral tissue and oral fluid over time. And then this concentration over time are then given to mathematical modelers to then see how we can then change doses to optimize these treatments in the mouth. So this is the mouth where we're collecting samples, and the red parts are where we're collecting our samples, collecting from the tongue, the back of the mouth, the tonsils. And as I mentioned before, we want to understand pH. So in terms of pH, we're measuring saliva pH by dumping some saliva in this pH machine to tell us what the pH is. To try to understand tissue pH, we're putting this end of this meter against the cheeks or the tongue to measure cheek pH. In terms of trying to collect tissue samples, where it's easy to get, such as the cheek and the tongue, we're using these dermal curettes, which we're using to scrape tongue and cheek tissue. And to reach the more harder to reach places, such as the tonsils and the back of the throat, we're using these flock swabs to try to understand concentrations in gingival fluid. So that's that fluid that's produced between the teeth and the gums. We stick this filter paper in it. And then we're also trialing this new method called VAMS, which is volumetric absorptive microsampling. And so what this is kind of a better version of dry blood spots, where you do a finger prick sample, and then you put that against that blood dry spot to absorb that blood. And so what we're trying to do in these experiments is trying to understand whether we can measure drug concentrations in these finger prick samples and how they correlate to blood that's taken from the vein, with the hope that if we can accurately measure drug concentrations using these finger pricks, then future clinical trials means that we don't have to get people to come into a clinical trial center. We can teach them how to take finger prick bloods and do it all at home, making it much easier and cheaper to run these clinical trials. So in summary, we've seen increased resistance to gonorrhea and other STIs. You need PK data to optimize treatments in the past, present, emerging. You need infection site-specific data and not the blood. And as I mentioned before, it's ideal to measure free drug levels, but you can't do that in the mouth and the rectum because you have to use these invasive microdialysis probes. So this is the stuff that you need to do to measure free drug levels. And you can't imagine putting that anywhere in the mouth or in the rectum. And traditionally, these are the stuff of putting them back in the leg or the back of the arm. So the idea is you need to try to determine what the best PD values for efficacy are. So what is that value at the time of MIC? What is that ratio of MIC to area under the curve? And the best way to do this is to embed pharmacokinetic trials into clinical trials. So you run a clinical trial, just add this extra arm where you take samples. The role of pH and saliva is uncertain. Emerging treatments such as olefluoride and jeopardized and offer some potential, but they're limited by small sample sizes. You can obviously optimize treatments to offset side effects. I think there's a need to move away from single doses. And in overall, PK/PDs are essential. You can use hollow fiber models or clinical trials, and obviously don't guess. And sorry for the disclaimer. And so I'd like to thank you for presenting and acknowledge that this work is supported by an ARC and NHMRC grant. Thanks so much.
Thanks so much, Fabian. We don't have any comments any comments or questions in the chat. Feel free to drop them in, folks. But also, feel free to unmute and ask any questions if you've got any.
It's very complicated, so I hope you were able to follow that.
It was hard work for me as a humble health promotion officer, but it was certainly very interesting. Thank you so much. Got a lot of thank yous and some claps.
Great, thanks. So I suppose the basis for this presentation is when you want to, if you've got an antibiotic and you don't think it's working, there's various ways you can change that dosing. You can split it, you can increase the dose, but you need to use these different measures to kind of work it out. Yeah.
Okay, great. Doesn't look like we've got any questions coming through.
Oh, Jane's got a question.
Oh, great.
Sorry, Ed, you might have actually answered this in your talk, so I do apologise, Fabian. But just going back to the ceftriaxone result, which had quite a low penetration into tissues based on one of your earlier tables, why does that work so well then?
Yeah, nobody knows. So the only other explanation I found that, if your tonsils only have 20% of drug levels at the time relative to blood, and there's none in saliva, I don't know. But if you look at the drug itself, it actually is what we call lipid soluble. It's actually able to dissolve into lipids or fats, and human cells are covered in fat. So these drugs don't really need any kind of active way to get inside cells. They can just passively cross these cells and get inside the cells and into tissue. So it's likely that this type of lipid solubility is likely to increase its penetration and make it work. And in fact, if you look at cephalosporins, that class of drugs that it belongs in, ceftriaxone is the one that has the highest kind of lipid solubility. So it's still a mystery as to why it occurs. Might be also because the MICs are much lower, so you don't need very much antibiotics to get into the oral space. But it's definitely the one that we use, and we're not sure why it works so well. So hopefully our PK study will understand a bit more about where those concentrations are going in the mouth.
Thank you.
Apologies. Questions from Dave. What are your thoughts on managing patients with poor medication compliance to move to I've lost it. Where did it go? multi-step treatments over stat doses?
Yeah, so that is a hard one, but let's reflect on chlamydia treatments at the moment, right? So now the treatment for chlamydia, doxycycline, you need to give that twice a day. You need to give that for 7 days, and most people manage to take that. And I think for gonorrhea, it's most likely that you're only going to need 2 doses. So it's likely 1 dose now, and then the other dose might be sometime that afternoon or something the next day. So I think there will be challenges in people who may not take that second dose, but I think we're just going to need to encourage them in a similar way that you would any other medication to enforce people to take those drugs as you would with any antibiotic. So I think that is just a measure. You could do SMS reminders. They've been shown that, for example, for HIV compliance medication, you can do apps to remind people. You can put reminders in calendars. But I think for gonorrhea, it should be okay because you only need to try to get that second dose in if we do 2-dose regimens. But at the moment, ceftriaxone is the one you give anyway, and that's an injection, and it has to be given by a healthcare professional. So it's likely for the current treatments we have now, that's going to continue to work. But yeah, that is a challenge. But I think the issue is with high doses, like I mentioned, if there's high doses, then lots of people are going to get diarrhea and vomiting. That's going to really potentially put someone off taking that second dose unless you can somehow optimize it to reduce those side effects.
And Fabian, are you happy? Obviously, we've recorded this session, so it will be available on our YouTube channel. Are you happy for us to make the slides available too?
Yes.
Great.
Yeah.
Thank you for that. All right, folks. I think this might be speak now or forever hold your peace, or reach out to us and we can pass any questions along, of course.
Thanks so much.
Cool. Thank you so much, Fabian. Thanks for coming, everybody. Hope everyone has a great end of the week.